All right, so thank you. So I'm going to talk to you about a project that's weeks in the making, weeks. So this is a lot of preliminary stuff, and it's actually related to looking at Malawi monads, which are one of those orphan taxa that Fabian had brought up. So Malawi monads are pretty cool. There's only about 3 formally described species, one from freshwater, soil, and marine environments. They have this typical excavate morphology, so they have this beautiful feeding groove and a vein in their flagellum, or flagella. And we actually have a near chromosome scale genome assembly from OASA, which we got last week. And what you need to know about these is that traditionally they're viewed as being part of Excavata because of their morphology. So oops, here is Malawi monads. Here are the metamonads and Discoba. So metamonads are things that like trichomonas that obviously is the disease. In Discoba, you have things like trypanosoma, which causes African sleeping sickness, along with Nigleria, which eats your brain. So these are fun organisms to think about. And this is from 2009, and so they were finding that Malawi monads are not necessarily within Excavata. But so long as you remove fast evolving sequences, then they do pop into Excavata beautifully with low bootstrap support in the trees. And the uncertainty by this is largely driven by just orphan taxa. And there are some reports and studies saying that there are keystone taxa to help resolve some of these relationships that are really deep and really obnoxious. And truthfully, I don't really think we have a really good answer as to where the hell Malawi monads go. So here they are near crumbs, which is that group that may or may not exist. And using a slightly different data set, a group has also found that, guess what, they actually are by metamonads. The metamonads are actually somewhere else that we thought. So we don't really know where they go. And this is great, but why should you care? And the answer is because we're eukaryotes, and we like to know where we come from. So a long time ago, this is a really cool paper where they essentially were looking at alpha proteobacterial markers from eukaryotic data sets. So metamonads are not there because they have mitochondria-related organelles. But the key thing to focus on is that you have Malawi monads near a member of chromes, and there is the root. So you actually can see that Discoba and essentially Malawi monads are on either side of where this root placement is, which would suggest that the last eukaryotic common ancestor of us was maybe an excavate. And then more recently, people looking at patterns of gene duplication and loss suggest, well, the root's actually with all Excavata, so metamonads and Discoba on one side, and then everybody else. If you do gene tree, species tree reconciliation approaches, you find that, guess what? It's actually between Episthocons and everybody else. And then if you decide that you're going to incorporate 3D models of proteins for your phylogeny, you say, metamonads aren't even a thing, but the root is actually within metamonads, or what we would traditionally view them as. And so when we think about Malawi monads either being near crumbs and Episthocons or near Discoba or near metamonads, everyone ignores Malawi monads because they don't behave. So where do they go in this tree then? It could be near the root. And so these are all really different approaches, and they do kind of add a little bit of their own flair to the story, I think. And they really highlight some things that we don't really think about too often, except for things like data quality. We're really obviously caring about that, especially in the advent of single cell techniques, where you can, and I've done this too, where you sequence yourself. It's embarrassing, but it happens. Also, filtering your alignments from bad sequences, bad columns, or removing non-homologous sequences, or portions of your thing because you have a largely disordered protein, can really impact these inferences. But also, one thing I want to highlight, and because I'm a very big skeptic, is I hate bootstrap values because you can get really good bootstrap values for poorly resolved parts of this tree. So we often overinterpret these things. And also, we usually hate gene duplications. It's hard to pick orthologs. So when you're picking one gene from a multi-gene family or one sequence from a multi-sequence family, it's really frustrating. But they're actually maybe not that big of a problem, especially when we start thinking about how gene tree, species tree inferences can help. And so people have actually been doing this in bacteria. So they've rooted the bacterial tree of life, whether you like it or not. It's kind of cool that they were able to do that. And so what I think we've been often doing is taking this classic phylogenomic approach where we have a set of markers that we like, where we develop a set of markers, and we just apply these big models, and we hope that we get the right answer, and we ignore everything else. And so what I'm thinking of is actually taking a step away from this and incorporating all of these things because they all kind of interact with each other. So classic phylogenomics is always going to be the mainstay. You have to do that using gene tree species reconciliation. Some people don't seem to like it, but I think that's where we need to go. Phylogenetic networks, it's always been kind of tossed around and ignored. We often ignore how genes evolve. We just hope that the models are good enough. And more recently, people have been using ancient linkage groups, at least to help root the metazoan tree of life, so animals. So is it really a comb jelly or a sponge or this placozoa thing? That's ancestor of all animals. Maybe it's ctenophores, who knows? And all this is the hope to actually resolve where Malawi monads ought to go. So this is the classic phylogenomic approach. This is the tree that NeLLi showed, the 317 genes. I think it's a great, excellent control. It's what we all recognize. We all know how to read them. We understand how they kind of work. The big thing is that they take time. And when we're discovering 1 to 2 new supergroups every year, that means that if you're not quick enough, you have to add them in, and then you have to do it again. And you have to hope that, somebody doesn't post something on bioRxiv and just go, la, 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 and you ignore it. That's my current opinion. Now, this has happened to us before. And so that's why I think, incorporating things like gene tree, species tree reconciliation approaches are nice, because they actually highlight the underlying discordance inferred from our species trees in these classic approaches. So this is, again, the same data set. And so here we have episthoconts, so animals and fungi. We have amoebozoans. That would be crumbs. It's not monophyletic. There's our cool Malawi monads, near and chiromonads. Here is where the traditional excavata are. So the things that African sleeping sickness and brain-eating amoeba, STIs like trichomonas, cool protists over here in SAR that are really diverse. I don't even know what that is. Oh, there's the red algae. So we actually don't see Archaeplastida anymore. And there's the green algae and glaucophytes. And here is Telenema picozoa with haptophytes. So we get lots of discordance that we're seeing from our species tree that's being highlighted here. And so this doesn't really help us with where Malawi monads go, but there's obviously no strong relationship with excavata that we can see from this one approach. So we're really seeing that there is some discordance going on. And so look at that. My background's in molecular evolution, so of course I have to go that route. Using this model called RELAX, it looks at selection intensity. So we always set a group to 1. So we're saying Malawi monads, whatever your selection is for any given alignment, you're 1. Everybody else relative to 1. And so if you go to 0, then you say, oh yeah, I'm an animal, I have stronger selection, I'm gonna be closer to 0. Or, oh yeah, I'm an animal and Malawi monads, I don't care, I'm gonna evolve faster. So I'm gonna be more relaxed. And so of the 245 alignments that I was able to do in a week, only 33 of them actually showed any significance, so deviation from 1. So if you're looking at amoebozoans here, they have more relaxed selection than our Malawi monads, which are right there at the red dashed line. And overall, most organisms, most of these deep clades are evolving more under a different selection regime than our Malawi monads. And what you really want is something that looks like this, where you're averaging about the same, because these alignments are showing that you have different selection regimes within a group. So if you're saying that Malawi monads are evolving more slowly than everybody else, your model is probably going to be a poor fit. And so we can identify genes that are evolving under similar selective pressures. That's actually what you want, because then everything's more homogeneous. The variation, the heterogeneity that we see is really going to be part of this problem that we're having. And what we can say so far is, at least from this pittance of alignments that I have, that Malawi monads are evolving under greater selection. We usually assume that it's purifying selection, but who knows? That's for another time. And so maybe the answer is going to what the chromosomes look like. So there's some structural information. So we often look at these genes as disembodied entities, but we know that compartmentalization of your genome is important. So if you're a trypanosome, the thing that kills many, many children, causes African sleeping sickness, your fast evolving genes go towards your telomeres, or in subtelomeric areas, or near centromeres. If you're an O-mycete, that kills potatoes. So that potato famine that we're all recognize, that's Phytophthora. It has a core genome that's really slow evolving in this gigantic wasteland of transposons and pathogenicity proteins. And so thinking about the way that these genes are passed down chromosomally matters. And so here's actually Trypanosoma brucea that kills a bunch of people, and Leishmania that also kills a bunch of people. And here we're just looking at patterns of macrosynteny among their chromosomes. And because I'm still developing this method, this seems like a good idea. They're really closely related. And here's the masa, there's our Malawi monad. And then the earliest diverging group now in the metamonads that we know of is this group called Anaeramoeba. And it's a bit depressing, or maybe that's a good thing. Because what we're looking for are genes that are kind of functionally linked together on similar chromosomes. So here, if we're looking again at Trypanosoma, this is what you want to see. You have a big chunk of genes here that look to be one unit that's well conserved. Again, this is over small time in a second group. And if I go to my Malawi monad and Anaeramoeba, you see nothing. So that's kind of fun. So this again is lending credence to me, at least, that maybe it's not part of metamonads, but also Anaeramoeba is kind of weird. So we can do this across other distances. This is all just sort of part of this early pilot that I'm kind of launching here. And so I think what we're really seeing, and that's fun to me, is that you get some phylogenetic inference that we may be able to build out of this. We know that it's working in animals, but that's only over a 600, 700 million year scale. And here we're talking about over a billion, and with different lifestyles. And if we're going to be truthful too, some things that are in the excavata have karyotype instability, so the chromosomes shuffle. Sometimes, like Giardia, sometimes it has 7 chromosomes, sometimes it has 14, depends on the day where you are. It gets really ugly. So from this pilot, what we're really hoping to do, what I think is going to work, is that we have this really neat set of approaches that do really help bolster one another. They're not actually functionally independent. They all add pieces to this puzzle to really help differentiate some of these things. And the beauty is too, is that we no longer have to rely on just one approach that is slow and is likely to be not totally accurate. But again, the limitation is that it is genome based, but with those keystone taxa that we can really largely focus on sequencing, I think this is where the field will go. So there's a lot of hope, I think. And what's fun too is that you can draw more inferences than just this is where this thing goes. We can start looking at the evolution of genome structures. Again, thinking about where we come from as eukaryotes. So how did the last eukaryotic common ancestor like blossom into the ridiculous diversity of things that we're finding? You know, when you go to the ocean, you just sequence a bunch of mags, and you're like, yeah, I found like 8 new supergroups. Like, I'm gonna cry. But you know, then we can start actually thinking about how these things function, and actually taking into account some of these other things we often overlook. So like the selective pressures in molecular evolution, evolutionary compartments, all of this are things that we often overlook, and we apply one blanket model for hundreds of genes from who knows what part of the chromosome they're even on. And we can do fun stuff like look at gene family expansions. Are there expansions underlying the pathogens in Discoba that kill bunches of people? Are there any uniquely shared genes that kind of tie some of these weird groups together that we can actually use as better markers? Right now, we're always saying you must have 80% taxon occupancy, but what if 5% of those genes are unique to 2 groups? And that would be a uniting feature that we could overlook because of the biases and how we've been doing it for decades. And so thanks for letting me go quick. I know everyone's hungry, so I was trying to go fast. I have to thank Kinsu for giving me the go-ahead to just start this ridiculous project, which is really fun. Sergey for being really, really patient and helping me with the relaxed stuff because it was a pain to get working. And Sally did a bunch of the original, actually, she did all of the manual curation lately, which is just, oh, I don't know how you do that. And Mario for arguing with me about gene tree, species tree reconciliation approaches.